everyone, my name is Jolaiya Tolulope Funbi. I'm presenting this video on behalf of African Helicobacter microbiota study group to have a standard operating procedure on the culture, isolation and susceptibility testing of Helicobacter pylori. Helicobacter pylori is a microaerophilic gram-negative organism. And when we are talking about a microaerophilic organism, it means that this particular organism requires a low level of oxygen to grow optimally. And this can be created with a mixture of 5% oxygen, 10% carbon dioxide, and 85% nitrogen. And in the laboratory, this can be produced with the use of a Funbi gel product from Thermoscientific company, and this Funbi gel gas pack will help in the production of 5% nitrogen. Here is the product. This is how it looks like, a gas pack that can produce a microaerophilic condition, 5% oxygen, 10% carbon dioxide, and 85% nitrogen. And if you look at this product carefully, we have 3.5 liters on this, depending on the gas jar that is going to be used. For a gas jar of this particular volume, this is like about 3.5 liters. This 3.5 Funbi gel gas pack will be used. There are bigger gas jar that will require about 5 liters Funbi gel gas pack, and this will be chosen based on the volume of the gas jar. This is another one. This particular gas jar will require about 5 liters Funbi gel gas pack. So microaerophilic condition that can be created by introducing this Funbi gel gas pack into the gas jar. So microaerophilic condition is required by H. pylori in order to grow optimally. Apart from this microaerophilic condition, H. pylori also is fastidious, meaning that it requires rich nutrients, and this can be created by the use of very rich agar base like Colombia agar, GC agar, and Brucella agar. But here in Professor Stella's laboratory, we use GC agar. GC agar can be prepared in a volume of 500 ml, and 18 grams of the GC agar will be measured on a weighing scale and measured into an autoclaving bottle, and 450 ml of sterile water will be added to the mixture and autoclaved. After autoclaving, the nutrients such as vitamin mix, this vitamin mix can be purchased commercially. For example, an Isovitalex product can be purchased, and it contains NAD, nicotinamide adenine dinucleotide, vitamins, ferric iron, and other amino acid and other factors. So it's highly rich. Apart from vitamin mix, horse serum or other animal blood products can be added, but horse serum has been in use in Professor Stella Smith's laboratory, and it's really giving us optimal growth of H. pylori. So a mixture of vitamin mix 1% and 9% of horse serum, meaning that in the 500 volume of preparation of a GC agar, a 5 ml of vitamin mix will be added, 45 ml of horse serum, and meanwhile 18 grams of GC agar have been measured into 450 ml of sterile distilled water. While autoclaving, a magnetic stirrer would have been introduced into the mixture of the distilled water and agar and autoclaved to avoid contamination. This magnetic stirrer will be autoclaved with the agar, and after this, it will be allowed to cool. The agar, the GC agar with the distilled water will be allowed to cool to 50 degrees Celsius to avoid coagulation of the nourishment that will be added. So after cooling to about 50 degrees Celsius, 5 ml of filtered sterilized vitamin mix, filtered sterilized horse blood, 45 ml, will be added to the GC agar mixture. And dense medium, this is a product of oxide. It's an H. pylori supplement, selective supplement, also known as dense medium, which contains vancomycin, amphotericin B, and other antibiotics to inhibit fungal growth or any other contamination. This will be dissolved because it's in powdery form. It will be dissolved with the use of sterile distilled water, about 1 to 2 ml, and then added to the mixture of the agar. And this now becomes a serum plate. The mixture will be poured under biosafety cabinet 2 after proper mixture of the dense medium, the vitamin mix, and the horse serum. So it is poured in the Petri dish under biosafety level 2. After pouring, there are bubbles. Flame will be used to remove the bubble, and then the plates will be kept in the dark to solidify. And while it's being kept in the dark to solidify, a clean napkin will be used to cover the plates in order to solidify and prevent penetration of light. So after solidification, if it's not ready to be used, it will be kept in the fridge. But if it's ready to be used, then what are the appropriate specimen to be used for culturing? The appropriate specimen that are recommended and optimal growth have been obtained from is biopsy. Although it has been reported that other extragastric samples have been used for culturing and H. pylori isolated, such as stool. Now, if biopsies is going to be obtained, biopsies can be obtained during endoscopic procedure, and Sydney recommended that 4 biopsies should be obtained during endoscopic procedure, 2 from the antrum and 2 for the corpus. In order to have optimal growth, a transport medium is required, and the transport medium that we recommend is Phytogen pylori transport medium. Phytogen pylori transport medium. While the biopsies are taken during an endoscopic procedure, it will be received aseptically and introduced or embedded into the transport medium, deep inside the transport medium, and movement of the sample should be within 4 hours, but it can survive up to 48 hours in the transport medium. Here is Phytogen pylori transport medium containing the biopsy obtained from the endoscopic unit, and the wire loop will be flamed in order to sterilize the wire loop so as to avoid contamination of the biopsy. The wire loop is now used to pick the biopsy from the Phytogen pylori and will be rolled over the surface of the serum plate. The 2 surfaces of the biopsy will be rolled over the serum plate, and if there are remnants of the biopsy, it will be returned back into the Phytogen pylori transport medium. If many samples are being cultured at the same time, there is need for proper labeling of the serum plate to differentiate from one biopsy to another to avoid mix-up. This plate is placed inside the gas jar. This gas jar is a 3.5 liter volume, and so a gas pack, Campi gel gas pack of 3.5 volume is introduced into the gas jar, and it is done rapidly to avoid the escape of the gas. This is done rapidly to avoid the escape of the gas from the gas pack. And after it is introduced into the gas jar, a paraffin can be used to tape the edges of the gas jar to make it airtight, to create an airtight environment such that the gas will not escape and oxygen will not penetrate into the gas jar. So this will be wrapped around, paraffin will be wrapped around the gas jar to maintain the microaerophilic condition, and then closed properly. Closed properly. Yeah, ready to be incubated at 37 degrees Celsius. Okay, so incubation will be done at 37 degrees Celsius for a minimum of 3 days and maximum of 10 days. So at the end of 3 days, if there is no visible growth, the gas pack will be changed and incubated further for 5 days. If at 5 days no visible growth, the gas pack will be changed and incubated for a maximum of 10 days. After 3 days incubation, the gas jar containing the plate that was cultured will be inspected. Then this inspection will be done under biosafety cabinet level 2. Okay, so the plate will be removed from the gas jar to examine for H. pylori growth. Okay, so we have visible growth of H. pylori, which are tiny translucent colonies, and they can be confirmed using catalase, urease, and oxidase biochemical test. For visibility's sake, I'm going to bring the plate closer to the camera. These are culture from antrum and corpus of patients from the endoscopic procedure. Okay, so from this culture, after confirmation with catalase, urease, and oxidase, the extinct colonies will be picked. Colony 1, 2, 3, and pulled. The extinct colonies will be taken on fresh serum plates. After confirmation of the presence of H. pylori on the plate using catalase, oxidase, and urease, the extinct colonies will be subcultured into a fresh serum plate. The wire loop will be flamed to prevent contamination, and just one colony, because here on the plate we have several colonies, just one colony, just one. Colony 1 on a fresh plate will be subcultured. The plate will be properly labeled for the purpose of identification. Colony 1. The wire loop will be flamed again. So we'll flame again. We'll pick another colony. Just one. Then subculture into a fresh serum plate. Colony 2. Depending on the number of distinct colonies desired, can pick 3, 4, and then for safety's sake, because this organism is very fastidious, you can pick pooled. That means you pick more than one colony and then subculture, and this will be harvested, and it will serve as a backup at -80. Peradventure, after a period of time, the different colonies are not growing anymore. So the different colonies that were subcultured will be incubated under microaerophilic condition, which will be created with the use of a gas pack. Like I previously explained, the size of the gas jar will determine the volume of gas pack used. For this gas jar, we are using 3.5 liters Funbi gel gas pack. So after incubating the subcultured plate for a minimum of 3 days, we have different pure colonies in the plate, which we are going to harvest into a freezing medium. This freezing medium can be prepared using Brucella broth, glycerol, and fetal calf serum. Following the manufacturer's instruction in the preparation of Brucella broth, to prepare 100 ml of freezing medium, 70 ml of the Brucella broth will be measured, and 20 ml of glycerol will be added, 10 ml of fetal calf serum will be added to make 100 ml freezing medium. So this will be used to harvest the pure colony, the colony 1 in a separate freezing medium, colony 2 in a separate freezing medium, and the pooled. The remaining colonies on the plate will be used for susceptibility testing and using e-test method. A 0.5 McFarland standard of H. pylori colony or isolate will be made for susceptibility testing using e-test method. And depending on the number of antibiotics that will be tested, if it is a 4-plate, take it for example for H. pylori susceptibility testing, the common ones are amoxicillin, clarithromycin, tetracycline, and metronidazole. So different plates will be prepared for different antibiotics for susceptibility. So for the first plate, we have amoxicillin, we have metronidazole, we have tetracycline, we have clarithromycin. This will be labeled with the identification number of each isolate. So a swab stick will be immersed into the McFarland standard of the isolate, and then streaked on the plates to be properly streaked on the plate for the different antibiotics to be tested for susceptibility. After streaking the organisms from the McFarland 0.5 McFarland preparation on the different plates, the e-test strip will be removed from the casing using a forceps after sterilizing the forceps to avoid contamination. So this will be removed and then placed centrally on the plates. The strip will be placed centrally on the plate. The e-test method is a quantitative method for susceptibility testing, and after putting the e-test strip on the plate will be incubated for a minimum of 3 to 5 days inside the gas jar at 37 degrees Celsius under microaerophilic condition using the Funbi gel gas pack. So after incubating the plates already that this e-test strip have been placed on with H. pylori isolates on, so we check for the inhibitory zones, and for this plate, amoxicillin e-test strip was placed on the plate after introducing the H. pylori isolates at McFarland standard concentration at 0.5 McFarland standard. The H. pylori isolates are not seen around the strip, so the minimum inhibitory concentration is taken at 0.016 microgram per mil. H. pylori is said to be sensitive to amoxicillin at 0.12 microgram per mil and resistant to amoxicillin at greater than 0.12 microgram per mil. For clarithromycin, H. pylori is said to be sensitive at 0.25 microgram per mil and resistant at 0.5 microgram per mil. For metronidazole, resistant at greater than 8 microgram per mil. For tetracycline, H. pylori is said to be sensitive to tetracycline at less than or equal to 1 microgram per mil or resistant at greater than 1 microgram per mil. The procedure of e-test or minimum inhibitory concentration using e-test strip can be carried out in duplicates or in triplicates before the final minimum inhibitory concentration is taken. And the frozen isolate, for emphasis sake, if anything is going to be done on it, probably the protein analysis, PCR, DNA extraction, or MIC test procedure, it must be done on ice. If it does, the isolate may not be viable anymore. Thank you.